Today we're going to talk about the digestive system. That we have the digestive system divided in two divisions. The first division is the gastrointestinal tract itself, and the second division, digestive system 2, will be on accessory digestive organs. In part 1, digestive system 1, we have three parts to it. We have part 1 has to do with the cells of the digestive system. Part 2 has to do with the oral cavity, esophagus and stomach, and then part 3 has to do with the small and large intestine. Enjoy. Digestive System 1, Part 1, Cells. Digestive System 1 will have three different parts to it. We will start with the cells of the digestive system, talk about the oral cavity to the stomach, and then part 3 will be the small and large intestines. But throughout the digestive system 1, we want to talk about the parts of the digestive tract, the four layers in the wall of the tube of the digestive tract, to identify the distinguishing structural features in various regions of the digestive tract, and to identify organ, region, and cell types throughout the digestive tract, whether we're looking at photomicrographs or the images that we see in the electron microscopic view or the light microscopic view. Now there's different cells and that's going to be throughout the GI tract, and that's what we want to do in this first part 1, is to talk about the cells throughout. In the beginning, we're going to be with near skin, and at the end at skin. So we will have stratified squamous epithelium at the beginning and at the end. Also, you have stratified squamous in the esophagus and the oral cavity as well. And it will change to simple columnar in the stomach, and then continue simple columnar all the way out until you get to the rectum. You pick up stratified squamous again. So lining the lumen, you're going to have two types of epithelium, either stratified squamous or simple columnar. In contrast, at the other side of the tube, there is another type of epithelium. We call it mesothelium, and that's simple cuboidal. Sometimes it looks kind of squamous too, but it's simple cuboidal cells located in through there. In addition to that, there's muscle. There's muscularis externa that we see there, and there's connective tissue. And in connective tissue, of course, you have blood vessels, nerves, and the immune cells would be located in there as well. So if you look at a stratified squamous epithelium that you have in skin, this is the dermis versus the lamina propria is what is the connective tissue below the epithelium in the digestive tract. And here we see a non-keratinizing stratified squamous epithelium on the lip and in the oral cavity. Here we see hair follicles out through here. This is the dermis, and a lamina propria on the other side. In the oral cavity itself, you still have stratified squamous epithelium. We can see that here and here, even over the tongue. There are different types of projections we'll talk about later on, but largely a stratified squamous epithelium in different forms. And another cell type here is the taste buds. So taste buds is another cell that interacts with the nervous system to detect one of the five senses, which is taste. Also, there are mucus and serous glands that will project out through here. This is a certain sarcovelate that pushes the fluid out of here, so you can use the taste bud to identify things. So other cells we see, these are mucus secreting cells with nuclei, flat nuclei at the bottom, and serous secreting cells. As you come down through the esophagus, you continue to have stratified squamous epithelium and lots of muscle. This is muscularis externa and the muscularis mucosa. In the muscularis externa, we have two types of muscle: skeletal muscle, largely in the upper region, and then as you move down through the esophagus, you have more smooth muscle, skeletal muscle we see there. In addition to that, there's nerve cells Here we see the ganglion cells. These are ganglia cells through here that regulates the contractions of these muscles. Still in the esophagus, we have stratified squamous epithelium, of course, lining the lumen, and then we have the muscularis mucosa that we see there, and here's the submucosa. And in there, we can see lymphoidal tissue. So we have lymphocytes, T and B lymphocytes are other cells that we have in there, and you would have macrophages and other things that are associated with development of the immune response to whatever antigens are coming down through there. In addition to that, so we have lymphoidal nodules. We also have the esophageal glands. Here we see the esophageal glands here and here, mucus type secretion that lubricates things that are being swallowed. So you come out from the esophagus with stratified squamous epithelium and get to the stomach, and the epithelium changes. From stratified squamous epithelium, the very same type as we had in skin, other than it's non-keratinized in the esophagus, to simple columnar. So we'll keep simple columnar all the way through the GI tract, and you will have different cells you see here, different cells in different regions. So as you come out of the esophagus right into the stomach, the first region is called the cardiac region. And the cardiac region has cardiac glands. So these are the glands here, mucus type secretions that coats any ingestion that may be belched back up regurgitated into the esophagus, to coat it to prevent the acid from digesting the esophagus. In fact, that's what heartburn is the regurgitation of contents and the burning of the esophagus. Once you get from the cardiac region, then you get to the fundic region. The fundus is the most active region, and we have different cells here. We see there's a host of different cells that we have here. The first cell on the very top in through there are the surface mucus cells. Surface mucus cells that we see there. And then what we have here are simple branch tubular glands. And so you just have pits. These are gastric pits in through here, and then you have a branched region for the two tubes. And in that region, that is the neck region. And so we have mucous neck cells in the neck region. We also have chief cells. Chief cells produce zymogen granules for pepsinogen. Then we have the parietal cells. The parietal cells which produce hydrochloric acid. And then we have another set of cells, the endocrine cell, the argentophane cell, the endoendocrine cell is basically an endocrine cell. So we have exocrine cells and endocrine cells in the stomach in itself. Here we see a toluidin in blue. These are the gastric pits. So you see here the surface mucus cells in through there, a lamina propria supporting the epithelium on the surface. These are smooth muscle cells in through here, blood vessels with blood cells that you can see in through there. But right in through this region is the neck region. The neck region is right through there. So the mucous neck cells would be right in through here. And then down here we had the fundus region, where you have the parietal cells, as you see there, and there'll be chief cells. So if we start at the top, we have surface mucus cells in the stomach, right in through there, and we can see the mucogen that is this is the luminal surface here, is the same as that. And you can see the droplets either here with toluidin in blue with light microscopic level, or at the EM level. Of course, you will still have tight junctions between adjacent cells to be able to make a lumen of the epithelial cells, and the lamina propria is down below there. We can see some smooth muscle cells there and some other nucleated cells, as we'll see more details in a minute. Also in the fundic gland, we have the chief cells. Here we see the chief cells. There's lots of rough endoplasmic reticulum at the base, and then it has these big zymogen granules which are released in the lumen. Here's the lumen right in through there. Again, all these cells would have to have tight junction between adjacent cells to create a lumen, for example. So here we have more chief cells, and then we can see argentavis cells, additional cells, and we have two different types of argentavis cells. Some make dark granules, some makes gray granules, and that is another cell that we can see there. So we start out with the surface mucus cells, and they are PAS positive because they have mucus. Also, the mucous neck cells are PAS positive too, because indeed they are mucus secreting cells too. So you have surface mucus and then mucous neck cells, and then you have the parietal cells, the endocrine cells, and the chief cells. Those are the cells that we see. So here we can see a chief cell with the big flocculent zymogen granules to be discharged. And then we have the parietal cells with lots of mitochondria in through there, very big nucleus, very big nucleolus. These kind of green ones we see are actually mitochondria. These blue ones are mitochondria as well, but these other guys are the chief cells. We also have our argentavis cells, which have smaller granules that are discharged in the lamina propria and hence into the bloodstream as opposed to in the lumen. Here again we see parietal cells. These are parietal cells here with all the mitochondria that's located in them. Another one right here. And then we have the chief cells with the big flocculent zymogen secretion that's in there. If we look at the chief cell, we can see a junction between adjacent cells, and we see lots of retinoplasmic reticulum and a zymogen granules that's going to be discharged on the lumen. Now, the zymogen granules of chief cells contain inactive enzyme pepsinogens, precursors which are converted in an acid environment to active pepsins, and pepsins initiate hydrolysis in a ingestion of proteins in the stomach. The digestion of ingested proteins is what it should have said. Argentavis cells have lots of granules at the base. We can see those here, and they discharge their contents in the base, but nevertheless they have contact microvilli projecting on the luminal surface to be able to interact with the lumen to detect signals to tell them to release their contents or not. And another one, a very important one, cell here in the fundic region is a parietal cell. And here we see the parietal cell with a big nucleus, a big nucleolus, and lots of mitochondria. And also another characteristic of parietal cells, it has a secretory canaliculus. That is, the lumen's up here, but the lumen extends down through the secretory canaliculus way down into the bottom of the cell. So whenever the cell produces hydrochloric acid, really it produces hydrogen ions and then chloride, which get together in the secretory canaliculus to make hydrochloric acid, so it can dump it here and not have to ship it all the way up to the surface of the cell. And so that's a characteristic of parietal cells. It's a big cell and has the lumen go down within the cell itself. The parietal cell also produces bicarbonate, which is a counterpart to the hydrochloric acid or a byproduct of the hydrochloric acid production. And also it produces intrinsic factor. Intrinsic factor is involved in vitamin B12 absorption of the gut, which is needed for red blood cell formation. So we have the parietal cell, big nucleus, secretory canaliculus, lots of mitochondria for transport across the membrane. However, if you have gastric atrophy, maybe caused by chronic inflammation, you get loss of the gastric glandular cells, and that may lead to loss of parietal cells, hence loss of intrinsic factor. And so you have the lack of vitamin B12 absorption if you don't have intrinsic factor. Vitamin B12 is a cofactor required for DNA synthesis of the erythroblast. So no intrinsic factor, no vitamin B12 absorption, no stimulation of erythroblast, and you become anemic is what could result from a gastroatrophy to occur. So if you come out from the stomach, the next region is the pyloric region. So from the cardiac to the fundic to the pyloric region, and here again you have the pyloric glands. I just want to point that out. Glands, these are glands, mucus type glands. So on either side, the beginning and the end of the stomach, you have cells that have a glandular content. So here, in case of heartburn, but in here also the coat, the ingestion, so you won't digest your small intestine, and that's what we see here is the small intestine. And in this so here's the stomach. We have a simple columnar epithelium of the different types of cells we just mentioned. And here in the intestine, we have simple columnar absorptive cells and goblet cells and argentophin cells. Of course, you have argentophin cells in both of these. But in either case, the stomach or the intestine, we still have simple columnar epithelium, just that the cells are different in different locations. So when you go from the pyloric or the stomach to the duodenum, the junction in through here, you go from gastric pits to intestinal villi and intestinal glands in through there, which will correspond to the gastric pit. But also we pick up these glands. These are the Brunner's glands, which is important as the first portion of the duodenum to lower the pH of the ingestion coming from the stomach. Now in the intestine we have several cells, and they all come from the same pluripotent cell. One of those is an intestinal absorptive cell with a brush border on the surface, lateral interdigitations of adjacent cells, lots of mitochondria for transport, especially in the apex and at the bottom of the cell, but that is a main absorptive cell. And then there's also a goblet cell. It has a kind of thin, small nucleus at the bottom, and then a big flocculent material here, which is a theca, as we'll learn later on, but that's a secretion portion of the cell. And then there's argentophin cells, which even though it touches the lumen, it discharges contents in the bottom, like any endocrine cell produces it in the connective tissue below and ultimately in the bloodstream. There's also in the intestine, especially in the small intestine, in the intestinal glands or the crypt of lubricant at the very bottom, you will see these pennant cells. And the pennant cells are involved in protecting the epithelium from bacterial invasion. Also, the duodenal glands, the glands of Brunner, also come from the same pluripotent cell. So we've got one cell here that gives rise to a host of different cells which characterize the epithelium, simple columnar epithelium that you see in the intestine. So here we see an absorptive cell with a brush border, lateral interdigitation between adjacent cells, the glycocalyx in through there. There's a goblet cell here, but the glycocalyx, there's a terminal web in through there, elongation of the mitochondria, lots of Golgi apparatus in through there, and the smooth and the plasma reticulum located in that region, as we'll see a little later on, and interdigitation between adjacent cells, and we have lots of mitochondria at the base for pumping things out, and up here for active transport on the surface. So if we look at the surface, the luminal surface, we have the brush border, and the brush border is uniform microvilli about the same length. High density of uniform microvilli is what makes a brush border, and we have the intestinal absorptive cells, and then also we have a surface coat here, glycocalyx coat, which is carbohydrates. Glycocalyx goes all around the cell, but it's amplified on the brush border, and it's partly involved in protection of the cells. So it protects against enzymatic digestion of the cells, and is active in the digestion process. The membrane of the intestinal absorptive cells has enzymes located in them, and so it can break down foodstuff right at the surface, so it can be brought right into the cell. Has several peptidases and you change peptidase to amino acids. It has disaccharides, disaccharidases for disaccharides to monosaccharides. It also has lipases for neutral fats to cholesterol and fatty acids, and also it has carbohydrate, a small amount of amylase is there as well, carbohydrates. So the various types of food stuff it can break down so that you end up with a product like, say, a monosaccharide that then can come into the cell. So in terms of the basic mechanism of absorption, you have active transport, and hence we have lots of mitochondria for certain things, for monosaccharides as well as amino acids. So they require energy to transport them through. However, the lipids are different. Fatty acids walk right through. Monoglycerides slip right through. Glycerol right through. And so once they get in there, then the triglycerides are resynthesized in the smooth endoplasmic reticulum. That's what we see here. And then you have the rough endoplasmic reticulum budding off protein, producing and budding off protein that goes with these, that makes these, after it buds off here, goes to the Golgi, and is added to the lipid is being coming through there, and then that produces chylomicrons. So chylomicron is a triglyceride covered with protein, and that's a chylomicron is discharged in a lateral border, comes through the lamina propria, through the central lacteal. This is the lymphatic, and that is in the intestinal villus. So here we see the intestinal absorptive cells, the brush border, the terminal web, and here is a smoothie or it's resynthesized in the triglyceride. The black thing is a triglyceride. So this is not just lipid, this is actually lipid component triglycerides that is in the smooth endoplasmic reticulum that's located at the apex of these cells. And here we see the lateral border between adjacent cells, which is where the chylomicrons are discharged. So you have lipid plus protein making a chylomicron discharge in the lateral border. And then it's discharged in the connective tissue, down in through here. And so in the connective tissue, you can see make the villus. The villus is composed of the epithelium on the surface and the epithelium and the connective tissue of the lamina propria. And here we see the central lacteal. So this is a lymphatic vessel that the fat goes from these cells in through there. We also have a host of other cells in here. We have lymphocytes in through there. We have plasma cells, which I can't see, mast cells I don't see, but you can certainly see macrophages. There's a macrophage right here. You can see the different components of degradation and phagocytosis that has occurred from these macrophages, but the other immune cells are there as well. Also, there's some smooth muscle cells that will be coming through there. I don't know if that's a smooth muscle cell, but we'll see some smooth muscle cells in a minute. Here we can see smooth muscle cells right there in the lamina propria. This is muscularis mucosa, the submucosa, muscularis externa. So this is smooth muscle in through there, smooth muscle in through there too, lamina propria, submucosa. In addition to that, we have some nerve cells in there too. This is the Meissner's plexus that are located in the submucosa that regulates the muscularis mucosa. So here we see some smooth muscle cell. This is a single cell. You can see the apical caveoli, characteristic of smooth muscle cell. There's part of a plasma cell with a rough endoplasmic reticulum shining through there, lymphocyte, and this macrophage with various stages of degradation. These are at the bottom of the intestinal absorptive cells. You can see the lateral underdigitation of the cells, and also you can even see the basal lamina going around through these cells. So the connective tissue cells in the lamina propria that you see there include smooth muscle cells and the immune cells as well. So speaking of the lymphoidal tissue, T and B lymphocytes that we see in through there, here they can be in the submucosa or they may be in the lamina propria, but the surface epithelium is a little modified. So we can see that the surface epithelium is a little different, and we call these M cells. These are M cells here they are antigen presenting cells, so they interact with whatever is in the lumen, and they pass it on to the dendritic cells, or like macrophage that may be there, other dendritic cells, which helps to pass the antigens on to the lymphocytes to get the antigen in the vicinity of the reactive lymphocyte. And so these M cells, or macrophage cells, or specialized epithelial cells in the mucosa, and they overline the lymphoidal tissue, which are known as the Prior's patches. The M cells selectively endocytose the antigens and transport them to dendritic cells and lymphocytes, which then migrate to the lymph node and then come back into the lamina propria to make antibodies against whatever the antigen that they were being exposed to at this time. Another cell is the Paneth cells, and we see those Paneth cells here with big granules, and they're located at the bottom of the intestinal glands, crypts of lubricant, as you might say. They are in animals and humans. The organelles indicate that it's protein. This is protein in through there. Those granules is eosinophilic, as you see, and it has lysosome capable of lysing bacteria, and that's what they do. So they're right at the bottom of the crypt. So it's bacteria slip down the lumen. They encounter the Paneth cells. They also concentrate zinc is another characteristic of these cells. So we can see those Paneth cells, lots of rough endoplasmic reticulum associated with protein secretion, and their discharge, their content at the lumen at the bottom of these intestinal glands. Now the Paneth cell granules release lysosome. There are lysosomes and lysosomal enzymes, hydrophobic peptidases, there's called defenins, and they break down the membrane of microorganisms. Paneth cells are important in the immune regulation of microenvironment of the intestinal crypts or the intestinal glands that we have. There's also goblet cells. There's a intestinal absorptive cell here, but these are goblet cells discharging their content. This is the theca here, the portion which is really where the secretion is itself. It's a unicellular gland in that it doesn't necessarily touch other goblet cells. It actually touches the intestinal absorptive cells. Flattened nucleus at the bottom just produces one content of theca and then discharges and dies. The whole epithelium is sloughing off every 6 to 8 days or so anyhow. And so these just produce and then they die and are replaced by other cells. And here we can see the goblet cell. These are the theca regions of the goblet cells there, and we can see it here and here. They have microvilli but not a brush border. And they do have junctional complexes with the cells beyond. They have Golgi apparatus, some rough VR, for is more of a mucus type secretion. Here we see the goblet cells here, intestinal absorptive cells, brush border, as we see of those cells. So we can see them. The nucleus is kind of thin in through there and dark. That's what we see in all through there. And then the theca portion being discharged right out through there. And here we see some terminal bars between adjacent cells. So why I have so much mucus, and especially in the large intestines, there's a host of mucus, is to protect the wall. And also it helps to keep the feces to adhere to each other, and it protects the wall against bacterial action. So not only degradation, but also from bacterial action, because you got a constant flow of this fluid. As a consequence, you have that so that the bacteria have a hard time getting a grip on things if more fluid is washing it away. So here we see the Paneth cells located right at the bottom of the crypts. In addition to the Paneth cells, we have the argentifin cells. These endocrine cells usually have golden granules located at the base, not at the lumen, at the base where they're going to discharge into the lamina propria. They're in the crypts. We can see those cells here. They're widely distributed throughout the GI tract, and they secrete various hormones like serotonin, which causes constriction. And so here we see the argentophin cells, even though they have the granules at the base, they interact with the lumen. So they interact with the lumen, and the granules will discharge in the base. Here we can see the esophagus and stomach and the different types. Some gastrin, secretin are located throughout the GI tract. In the stomach, there's a moderate amount common, rare common in the duodenum, duodenum, ileum sparse, and abundant in the appendix. And basically what it is one cell talking to another cell. So the stomach produces pancreatic glucagon to talk to the liver. Okay, and the pleuric duodenum makes a hormone that interacts with other endocrine cells. Is what happens. Here we have gastrin to tell the gastric secretion. So this is a pleuric and the small intestine telling the stomach to produce more gastric juices. Here's the small intestine using cholecystokinin in one of the granules to tell the gallbladder to empty. So this is a communication system between one region and another. Small intestine to tell secretin to produce more pancreatic juices. So this is a small intestine talking to the pancreas, and that really does that by these hormones that are in the argentophin cells. So here with muscularis externa, muscularis mucosa, submucosa. This is the mucosa in through here, which is the epithelium and the lamina propria muscularis mucosa. That makes the mucosa. And here we see the central lacteal going in through there. In addition to those cells, the goblet cells and the intestinal absorptive cells, we also have nerve cells. This is Meissner's plexus in the submucosa, and the Auerbach's plexus that's located in the muscularis externa. So if you come from the large intestine, there's lymphoidal tissue in through there as well. And then you come to the anal opening and you change from simple columnar to stratified squamous again. So from simple columnar to stratified squamous at the anal opening, and lots of goblet cells in the large intestine that we see, and that's where we at one end, and as well as the other end, you have a mucous cutaneous junction. So it's a good place to end right here at the end of the GI tract. We want to thank a host of books, websites, lab guides that provide images. The original images, which is very good, are all from these books and other sources. I did not make them myself. This is the end of Digestive System 1. Part 1 has to do with cells. Next time, we will talk about the oral cavity, esophagus, and stomach.